Now, here's what that looks like. So if you look at all cancers, this is how it comes out. It turns out chromosome translocation, where you put two chromosomes together improperly, form a big part of the problem in most cancers. And that's because when you do this and you do it improperly, what you generally do is put a section of DNA that makes a gene turn on in front of a DNA that should normally be tightly regulated, and as a result, the gene is on all the time, telling the cell, divide, divide, divide. And then you've got symbol and somatic mutations. These, I think, are mutations that don't actually change the amino acid coding sequence of the gene. But if these happen in a regulatory region, they are significant. Symbol mutations change the coding of the gene. Now, those of you who know the redundancy of the code know that that's a potential problem, or that potentially, if you know that amino acids can substitute for one another but not change the structure of the protein, that can be kind of dealt with. But if it changes a critical amino acid, such as a cysteine, it can drastically alter the folding and formation of the protein. Then these are all mutations that will, frameshift mutation stops the transcription of the gene before it even gets started, and then nonsense stops it. Frameshift results in it reading differently. Deletion results in you got a big chunk of the gene missing, and splice site effectively does the same thing. So all of these basically break proteins. Breaking proteins changes the cell cycle. Now, ask the fundamental question. As discussed before, they're only looking at 600 genes in the genome in finding these mutations. What happens if the genes that are not involved in the cell cycle, they aren't involved in cell division, break? And so that's where we get into aging. All right, so one of the things, about 20%, it depends upon the cell type actually, but 10 or 20% of the proteins that are synthesized in a cell go through the endoplasmic reticulum because they're supposed to wind up on the surface of the cell, ultimately. Now, over time, Michael Lieber has estimated that by the time you are 70, you may have several thousand repaired double-strand breaks in each cell. Now, if you repair those breaks in the junk DNA, who cares? But if you have to repair those breaks in a regulatory region or a coding region of the protein, there are consequences. So one of the consequences is you get frame-shifted genes or proteins which will fail to fold properly, and that results in proteins on the surface of the cell which are potentially foreign because they don't look like they looked like when your immune system was getting trained. And as a result, you get increases in inflammation, which is a hallmark of aging. Or they may just sit there and be non-functional. And so whatever they're supposed to do, they can't do it anymore. An example of which would be a hormone receptor. Testosterone therapy may not work if the testosterone receptors are non-functional. So there's all kinds of consequences. Now, as it turns out, the cell has a way of detecting that this situation is going on. For example, you go out and eat a pound of sugar. Well, that's going to result in your pancreas having to produce a lot of insulin. Well, the pancreas may not have you wonder why the pancreas is one of the bigger organs in the body, and that's because people go out and eat a pound of sugar. And there is an endoplasmic reticulum stress or unfolded protein response. So if you're basically shoving proteins into the endoplasmic reticulum as fast as you can, and they are not folding properly because they've been mutated, then this response gets activated. In eukaryotic cells, you've got 3 genes, triple redundancy here, to make sure you turn that response on and go after. And what it does is some of these are transcription factors, and some of these are inhibitory factors that will turn down protein synthesis. So one, protein synthesis stops for most genes and waits until the stress response has gone away. Then it also increases the heat shock and chaperone protein production because maybe the endoplasmic reticulum hasn't got the capacity to do this. And then it primes the cell for apoptosis. This is critical. If you are shoving enough proteins through the ER that are trying to fold properly and don't, then the cell will ultimately commit suicide. And the reason for that is simple. Why should it be consuming energy synthesizing proteins that it can't do anything with? Mitochondrial stress, there also is one of these stress responses for the mitochondria. And so there's less direct feedback, but it's got a whole set of genes involved in trying to clean up the mitochondrial stress because, as Aubrey said yesterday, it's like 1,000 of the proteins end up going to the mitochondria. And it would make sense that if you get these two compartments, the endoplasmic reticulum and the mitochondria, they should both be able to detect when things are messed up and give a feedback to the transcription. Now, the consequences are that if the mitochondria are only just kind of screwed up, you'll get increased free radical production. You may have insufficient ATP for cellular activities. You may have to increase the number of mitochondria. But if you can see what's going on here, first you have to synthesize the proteins, then you have to put them where they're going, then you have to take them back out and recycle them, which is what the proteasome is for. Well, that ends up increasing the requirements of the cell for ATP, which is going to feed back and cause the mitochondria to produce even more free radicals. So you're in a vicious loop here. You've got to repair the damage, but repairing the damage requires more energy. More energy generates more free radicals, and the free radicals generate more damage. That's not good, but that's the way the system was designed probably 1 billion or 1.5 billion years ago. Somatic mutations cause, this is the summary basically, this is the kind of like the punchline. Whoops, sorry. And you get protein misfolding, you get aggregation, which was discussed yesterday, and you get inflammation. Cells respond with diminished function, they slow down basically. Who would not agree that aging is a phenomenon of cells slowing down? And so the recovery is recycle the proteins, increase ATP production, increase mitochondrial activity, and therefore free radicals. There's the vicious loop right there. It's like I need more ATP, generate more ATP, but there are consequences for that. Plan B, commit suicide, okay? Plan C is, okay, well, I committed suicide. What you'd like to have each cell do is commit suicide, send me a replacement from the reserve capacity. This is why being larger is a good idea, because you've got more stem cells in larger organisms than you do in small organisms. The result is gradual systems failure. So, can you produce a grand unification? Free radicals are the source of the problem. Somatic mutation is the consequence. And so this was generally cast out of gerontology, but it turns out that now we can put it back in as being a legitimate problem. Distance differentiation, if the damage to the genomic DNA is causing mutations that are in the regulatory regions, the cells will lose their idea of who they're supposed to be. And the whole architecture that's on the wall back there begins to come apart. Protein synthesis, well, you're able to see how the cell is spending an increasing amount of its time shutting down protein synthesis as the mutations accumulate, which means your protein synthesis abilities decline. And you can kind of link the mitochondrial theory of aging in here because bad proteins going into the mitochondria not folding properly help the mitochondria generate even more free radicals than it normally would. And then what's come in the last few years is the fact that the Hayflick limit we heard about yesterday was due to the telomere shortening largely. But then there also is the question of, is there a stem cell limit? Are the stem cells aging too? And for many years it was thought that stem cells do not age. Stem cells just age slower. Stem cells are kind of like on hold. But then when you need them, you go to get them, and it turns out if they've accumulated sufficient damage, they can't do what they were designed to do. So the fundamental reason, if you want to sum it up in a one-sentence quote, why do organisms age? Evolution has no do-overs. Because it made decisions very, very early on in the process of the structure, the architecture of cells and how you get energy. And if a programmer were to look at that and say, you did that? Boy, is that a really stupid idea.